Hello, welcome you all in the UniProtKB database visit. For visiting the UniProtKB database, simply you have to open any browser. I will suggest Google Chrome browser is a more easy to use browser and non-hectic browser. It's not a paid promotion of Google Chrome browser, but it's a non-hectic browser. Okay. Instead of that Microsoft's browser that it contains lots and lots of advertisements. Here you can't see any advertisement or anything, so I generally prefer it. Here you have to type UniProt then KB. Okay, it's not a case sensitive, you can type in all caps or in all smalls. So this kind of thing open, you have to select, you have to click on first ever site UniProt. Now just wait for 1 minute and it will be load. So this is the basically entire uh, homepage of the UniProtKB. Okay, here you can see the option of blast, align, peptide search, mappings, parql, everything. Blast and alignment that we'll see in the next week, so you need not to worry about it. Here also they have collaborated with the Google DeepMind, so for the protein structures. And here you can see proteins in the UniProt knowledge base, that is UniProtKB. Species proteomes, protein clusters in UniRef, sequence archive in the UniPrac. Okay, we just have to focus on UniProtKB as of now. Here you can search what you want to check, but I will suggest you at this moment just go with the UniProtKB. And here I will go with the simple insulin, human insulin. Okay, insulin, and then I will type homo sapiens. Okay, homo sapiens, and that's it. Here you can see the list. About 1904 results are available. Here you can see this in the card format, so this is called as the card format. And here you can see in the table format, this format is called as a table format. No matter in which format you want to see, you have to choose it. Now this P01308, this entry is for the human insulin, simply the human insulin. So I will open the first one. Here you can see the protein name, the gene responsible for this protein, INS gene, status, then organism, from which organism it is taken, amino acids, how many amino acids are there, protein existence, and annotations. Now here you can see the function of this protein. So insulin decreases blood glucose concentration, it increases cell permeability to monosaccharides, amino acids, and fatty acids. It accelerates glycolysis, the pentose phosphate cycle, and glycogen synthesis in liver. Here you can see the GO annotations, ontology annotations, and GoCam models. Okay, we'll visit that one also. Now here you can see the just don't change this one. Okay, here this option shows at which level you want to see. So if you want to see as the eukaryotic cellular processes, you can select this one. uh, okay, no problem in that. Now here all annotations, all molecular functions. So here you can see the all molecular functions. Now just see the video on the gene ontology module 2.4b. Okay, you can see the I button before the means above the this video. So you can directly land on that video. So see the gene ontology video. What is the gene ontology actually? Where you will get. So these are the molecular functions. Here you can see this gene is involved in the catalytic activity, then protein binding and molecular regulator activity. In biological processes, you can see this gene is involved in the generation of precursor, metabolites, and energy. Then here you can see the DNA templated transcription. This gene is involved in the this one lipid metabolic process. So all these things you can see, and here you can see all the biological processes, molecular functions, and everything. Also, you can see about the uh, cellular locations. Okay, now here you just have to scroll down, and here you can see all these things in a list format. Okay, now here you can see the pango where these annotations means you can check for the uh, pango database. Now enzyme and pathways database, you can check the pathways and everything. Protein family or group databases, so you can go on allergome, and this allergome you can check about this. Now all the information about this thing, in which organism this particular thing we can see, and all this taxonomical lineage of this particular Homo sapiens is given. Accession number where it is you can find proteomes and everything we can check. Subcellular location. Okay, here you can see the secretion. Okay, it is secreted outside the cell. Now you can see about in which diseases. So insulinemia, hyperinsulinemia, type 1 diabetes mellitus 2, diabetes mellitus permanent neonatal 4. So in this particular diseases you can see. Now I will go little bit below how it is processing and everything. Okay, interaction. Just 1 minute. It is loading here. Okay, so you can check about the interactions, how it forms the protein-protein interaction. Means how it is forms the interaction with another proteins. Okay, uh, and also the 3D structure of this protein. Okay, so it is loading a little bit. Now, without wasting so much time, I will just go in the top of the page. Now, see, you can visit this database, and I will suggest you, I will strongly recommend you to visit this database and see this particular protein or any protein of your choice or any gene of your choice. Now I will click on this go cam models. This is little bit interesting model here. Here you can see now suppose I want to see about the insulin mediated activation of ARF6 RAC1 axis, and here you can see how this thing is involved or how this thing is related with each other. Now see, just see this arrow. Now this green color dotted arrow is there. That means indirect positive regulation. This insulin indirectly uh, positively activates the PRKCL. Here you can read about this one. This PRKC will KC I or L, I guess it's a L. It directly positively regulates or activates the APPB1, and this actively directly activates the ARF6. So this all informations is here. You can check each and every one, all the activities. Now I want to see about this one: insulin synthesis and delivery to skeletal muscle promotes glucose uptake. Now for this, I will just you have to use the mouse to zoom in and zoom out. You have to scroll the mouse. Here you can see glucose, and here you have to take the information help of this table. Here it means input of glucose gives input of SLC to A1, which has output as the glucose, means SLC1 glucose acts as an input and output, and it positively regulates the PDX1. Here PCSK1 positively directly activates INS insulin, and this PDX1 indirectly activates positively regulates the INS1. So this is how you can check. Okay, so and here all the activities you can read from here, processes and activities, everything. Now the same thing is here, so not to see about then variant viewer, what are the variants here. Okay, so likely pathogenic or pathogenic, these are the other variants of this one. Okay, this means what other types of uh, things are available with this protein, uh, and what is their relationship with other, means if we change this one with this one, what will happen. So if we change this one with the G, then it will it will do some consequences. If we change this R with G, then it will do some pathogenic or likely pathogenic reaction. Feature viewer, here you can see the features. Here all the features are there. Genomic coordinates. Okay, nothing is there. Publications, you can see about the other, all the publications, all the possible publications uh, mentioning this gene. Some external links, means links to the other sources and all. And then here you can see the history. Okay, so all the history you can check. So that's it about visit to UniProtKB, where you can go and check all the gene ontologies here and the information about the protein here. So that's it for this demonstration. We'll meet in next one small demonstration regarding the gene ontology database. Thank you.